Welcome everyone. So I'm going to give an overview of fungi I've been finding in the Miami area. So we'll give a little introduction and then go through a lot of photos and species that I found in Miami, and then a little bit about the why I'm doing this at all. So three great kingdoms of life that we typically think of are animals, plants, and fungi. All right, so I kind of want to draw some parallels to animals and plants when we talk about fungi as their own kingdom. So if we think about the animals of South Florida, we know them fairly well, right? So we can say Miami tiger beetle, right, around the zoo is endemic and it's rare. The rimrock snake, endemic and rare. The Cape sable seaside sparrow, an endemic subspecies, or the Strauss's swallowtail is rare in Key Largo area. Right, so we've looked for these things, we've looked at animals a lot, and we know that they're rare. And some of them are endemic because we found them nowhere else. And plants similarly, right, we've studied them very well. These are 4 endemic plants in the Miami area: lantana, melanthera, aster, consolea, cactus, and this poinsettia, now euphorbia. Right, so we know our plants and animals fairly well. So for fungi, there's kind of this ratio that's a good estimate of species richness of fungi. So for every 1 plant species, it's thought there are 4 to 6 species of fungi. Right? So if we take into consideration 1,200 native plants in the Miami-Dade County area, from mosses to flowering plants, so on, 1,200, and we apply this fungal ratio, we're saying maybe there's 5,000 or up to 7,000 native fungi species just in this county. Right, so, but there has not been a lot done. So here I'm highlighting maybe 40 publications that have focused somewhat on fungi in the Miami area for thousands and thousands of species. So there's you can find probably 40 publications on Strauss's swallowtail, or maybe 20 or so alone. So there's been a few publications on freshwater fungi in the Everglades and a smattering of other things, but not much else. So, what do we have here? So that's what I'm kind of trying to find out. But I want to remind you that fungi are very diverse, especially at the microscopic level. All right. I don't even see my cursor. I see. So, okay. So especially at the microscopic level, fungi are very diverse. All right, so 90% of the species are microscopic. We don't really see them unless we use a microscope. So that's what's mostly in Miami as well. I've been focusing on macroscopic fungi. So the ones that I can see, just walking in the woods, ones that form mushrooms, for example. Lichens, they're at the bottom right. That's a pretty popular lichen in Florida. Those have been studied pretty good in Florida as far as fungi go, but almost nothing else. So I've been trying to focus on the larger mushrooms, non-lichenized fungi. These are the main areas I've been looking at. So in the west, at the Everglades, that was one of the first permits I got. Key Largo, so a little bit south. The redland hammocks, so like Castello hammock, a lot of nice hammocks in the redland. Urbanized areas and natural areas around urbanized areas like Matheson Hammock and Deering Estates are heavily surrounded by the city. And then even Key Biscayne, so even beaches have some interesting fungi we'll see. So my favorite fungi are mycorrhizal fungi. Maybe it's because the botanist in me and the sort of teamwork at play in nature, but 90% of plant species are mycorrhizal. So they're in a mutualistic association through their roots with fungi. So at the bottom there, the fungi, the N and the P, fungi are very good at getting nitrogen and phosphorus minerals in the soil because of their very high surface area. So like all these white hyphae on the left there, right? They're absorbing all those minerals, and then they're giving them to the plants. See, those hyphae are surrounding the plant roots. And then the tree or whatever plant, could be grass, could be a pine tree, right? They're getting all that carbon out of the air, the carbon dioxide, and that's surplus to them as well. So they can give up to 25% of their carbon will be given to fungi in this mycorrhizal relationship. So most plants, again, do that. But again, most fungi are microscopic, so about 88% of plants species that have these mycorrhizal relationships are mycorrhizal with small fungi, microscopic ones that complete their life cycle in the soil. So again, I'm kind of interested in the big mushrooms. I mean, I'm interested in them all, but the easiest thing to study at this point is so far the mushrooms. So only 2% of plant species are mycorrhizal with mushroom-forming fungi. So they're exchanging nutrients in the soil, and then these fungi, when they reproduce, they form big mushrooms. All right, so only 2% of plants have these relationships with mycorrhizal mushroom-forming fungi. And in South Florida, our most important ones are the oaks, the pines, Pisonia does some weird stuff with some mushrooms, and then cockaloba. So cockaloba, the genus of sea grape and pigeon plum. It's a very Caribbean genus, so it's kind of been one of my favorites to gravitate towards. So if I see a pigeon plum or a sea grape and it's been very rainy, I immediately go right towards them, and I'm just looking underneath to see if there's fungi, mycorrhizal fungi associated with them. So kind of my first story, my first species, just begins with me moving from Tampa down to Miami. So that was July 2018. I didn't know much about the area. I had a lot to learn. And so this is my kid down there. He was 1.5 at the time. So I took him to the pediatrician, my wife and I, and then we went to get lunch. And so the story of discovery of this fungus is just me going to lunch. And the thing is, he's 1.5, so you can't strap him in a chair without food or something to do. So I usually order, and then I walk around outside. You know, outside they're always distracted, and they really get entertained, these young kids. So I'm just walking them in the stroller, and then on the top right there, you see a big tree trunk, and there's little white lines in the soil. So that's kind of all you're seeing. And that's kind of the first cool, fun fungus, mushroom, if you will, that I saw down here. So in the bottom right, that's a kind of a closer image of it. So it's just these gray, whitish, or even blackish coral-like fungi. So they kind of branch like coral fungi, like corals, and they call these coral growths, coral fungi. So that's a closer image. This is one from the Everglades. This earlier one's from Pinecrest. That's the first one I found was in Pinecrest. But I also found it in the Everglades once I got permission to go there. And now I see this fungus everywhere. But I can see why it might be overlooked easily, because it kind of looks just like it's, I don't know, something decaying, this grayish, blackish color. But it turns out those observations in 2018 were the first ones since it was described 76 years prior. So kind of fascinating. But since I've seen this, I'm almost tired of seeing it now because it's such a common fungus. Almost every sea grape pigeon plum has some of these around it, even planted ones like the ones I saw in Pinecrest. So they really adapt to disturbed soils even. In the bottom right there, you can see a dried collection. So that's a collection from the 1940s by Rolf Singer. And Rolf Singer is one of the most famous mycologists who studied mushrooms, macrofungi, big mushrooms that we can see with our eyes. So that's just kind of cool. He's a really historical figure in mycology, and he spent a few months here in the Miami area. And he's kind of the only person that's studied mushrooms in the Miami area. And then on the left there is Aryan Farid. So I just want to mention him. He's been helping me a lot. He's a grad student at the University of South Florida, and we study the fungi a lot together. Just like, well, this is Clavulina floridana. I'm sorry I didn't say the name. That's the genus species of that fungus, which is, again, very common around these cockalopas. And chanterelles are extremely common around cockalopas as well, both species. At one time, this was called a different species, Cantharellus cibarius, I think. So this species that associates exclusively with cockaloba was only figured out, if you will, 2016, partly due to DNA work. So it's DNA that really showed us that this is an independent lineage of chanterelles that is exclusive to cockaloba. Before that, they basically used a name that was described from South Carolina. And of course, there's no cockaloba there, but they looked similar, kind of a bright orange color. But now we know they're very different. They're not interbreeding lineages whatsoever. This is a different species. And again, it's very common. I see people pick these and eat them quite a bit. And that's one of the few very recognizable mushrooms in the area that people feel safe to eat. So there's so many mushrooms that can be found under cockaloba. So here's another one. Here's out in the Everglades, again with my colleague Arian. So a lot of my mushroom discoveries are just kind of happenstance. So in the bottom left there, someone's tying their shoe, and that's how I found this one. I mean, so many times it's just because I stop, that's when I find the mushroom. It's not like I have a sixth sense or something. It's just I stop and suddenly I'm taking in the surroundings and looking closely. And this thing's kind of tiny. It's only a couple inches high, and it's kind of gray, so it's difficult to see until, again, I got down on my knees and really looked at what was going on the ground. And it's, again, in association with pigeon plum. I've never seen it with sea grape, but it's a bolete. So it's the genus Fistulinella glowiocarpa, a bolete. So bolete, B-O-L-E-T-E, describes a group of fungi that usually have a typical stem and a cap, but underneath the cap are pores, not gills. So like the chanterelle has more gill-like spore structures underneath. Boletes have these pores, usually have pores. So that's a very important group that are mycorrhizal with a lot of different trees. And this was the first documentation of it in Florida. So Rolf Singer had not come across to this either. And then going to the beach, there's even beach fungi. And here I'm looking at sea grape. Right? So there's a lot of sea grape just beyond, the high tide, or a little bit further past in the coastal strand. So this was just a month ago. I was with my daughter, so now I have 2 kids. And family outings are a lot of my discoveries of fungi. It's like I can't go out by myself a lot of times. This was a Saturday and it had rained a lot, so I thought, I really need to take this opportunity. I don't know if it's going to rain on the beach again, so let's go see what's out there. So I took my daughter with me and strolled around. She's trying to grab everything she sees that I have my hands on, and found 2 very cool mushrooms popping up under sea grapes. Again, so these mushrooms are just exchanging nutrients under the soil, and then when they get enough rain, they pop up and sporulate. So the first one is Amanita there, popping out of the sand, and the second one is the genus Russula. So Amanita, it's a very big genus, about 600, 700 or more species, if I remember. And the gills, right, that's where the spores come off, they are free from the stem. So they don't really connect directly to the stem typically. So that's a helpful character for Amanita, and the gills are typically white, a very white color. And I don't know the species yet. I have some guesses, but I want to do some DNA work to hopefully figure that out. On the right there is Russula. The gills actually do touch the stem and connect to it. It's a bit hard to see. And the stem on a Russula tends to be pretty stalky and stout. And the stem is very crumbly. So if you feel it and you squish it, there's no rigidity to it. It's not a very strong structure. And the discoloration in that Russula mushroom is from maggots. So these mushrooms, there's little insects, flies and whatnot that lay their larvae, eggs in there, and the larvae develop, and they start eating through these quickly. So that's not the best specimen, but it's the only one I found of that species that day, so I did keep it. And then I could go on and on about cockaloba, but here's a nice overview of the many species I found just in South Florida under sea grape and pigeon plum. All right, so if you go to the Caribbean, there's even more species or different ones that you'll find under the genus cockaloba. So on the left, I have Clavulina floridana. That was the first one I showed. And then there's a white one that has a coral-like growth form. Another Clavulina, different species. There's probably 2 chanterelles. So just yesterday, Deering Estate, I found a different chanterelle in the bottom left there. It kind of has a yellowish, grayish tone to it. I don't know what that one is. I was that was unexpected. I've only seen that one so far. In the middle there are a whole bunch of boletes. Again, have pores underneath. So there's Fistulinella xerocomus that has yellow pores. So that's another one. I have guesses on the species. There's a lot of work to really figure out what the exact species is or if it's undescribed. In the middle there is Neoboletes with the pores. And then the bottom, completely different growth form, the genus Scleroderma. So these are kind of called puffballs, in that they start forming a ball in the ground, and then they eventually peel out, and their spores just kind of form a powder that puffs around into the air. So that's mycorrhizal as well with Cockaloba. The top right, there's 2 Amanita species I found at least. And in the middle right, this fuzzy looking thing, kind of a weird spongy-like mushroom. I had no idea what that was when I first saw it, but turns out it's another fungus that's mycorrhizal with Cockaloba. So all these different lineages, different growth forms, evolving these relationships. That's very cool. And then the bottom right, there's again Russula. So there's a lot to find under Cockaloba, and I really, really enjoy seeing that species out in the wild, or even when people plant them, because it gets me excited about finding mushrooms underneath. All right, so we'll move on to genus Pisonia. I just got one mushroom I'm going to show there, but there's one species that's a shrub like on Big Pine Key, the lower keys. But the most common species of South Florida, right, it gets onto the mainland as well, is Pisonia aculeata. So that's there on the top right. It can form these very big vining, scrambling thickets. And then the bottom right there is its flowering. And they typically have thorns on them, and so it's not fun to walk through them. So I get tangled quite a bit. But they form mycorrhizal relationships with things that form above ground sporulating structures. So you could call this a mushroom, I guess, but this is the genus Thelephora. The bottom left there is the name. So that's a very common group, commonly mycorrhizal with Pisonia. It's not necessarily a pretty one, but it's just, for me, it's very cool to find it. Right? So there's probably at least 2 different species I found so far with Pisonia. These 2 are both from Key Largo, but I think they're different species. And again, the DNA work really helps to figure out what these are. I'm pretty sure they're undescribed species. No one's really worked much on Thelephora in the Caribbean at all. So it's kind of exciting. But again, a lot of work to actually figure all this out. And then the genus Pinus. So that forms a lot of mycorrhizal relationships with mushroom-forming fungi. So here we have, of course, pine rocklands, which burn a lot. So in these burning pine rocklands, we really don't find a lot of mushrooms. And that kind of makes sense because the leaf litter is being burned away. There's fires rolling through. So a lot of the fungi seem to be adapted to be underground, underneath these fires. So this was one I again found yesterday at the Deering State. So they look like little brown dung pellets almost. And they're kind of partially buried in the sand. And I just got lucky enough to spot a little colony. So these are called false truffles. So truffles grow underground. That's a totally different group of fungi. This one's the false truffle, which is actually related to the boletes. So it's kind of a bolete that just evolved to only form this globose structure. The spores are all inside. So that's kind of a common theme for fungi in heavily burned areas like pine rocklands. Whereas if you go up north, where you get sand hills or other hammocks that have pines in them that don't burn a lot, you'll get different mushroom-forming mycorrhizae. And then the lovely oak, we only really have about 2 species in South Florida. One that forms trees, the live oak. You can find a lot of mushrooms under live oak. A lot of ones. So here's just boletes, the group of boletes. Top left, hortoboletes. That's a very common one in under planted oaks, so disturbed areas, it doesn't mind. The bottom left is actually a bolete. That's the genus Philoporus. It just evolved to have gills in that group, whereas most have pores. And you can see these bluish stains on both of those boletes on the left. So a blue stain is something when you bruise the fungus, some of these boletes do. They discolor, and that's a helpful character for identifying them. On the top center, boletes. That also has this bluing discoloration when you handle it or bruise it. The bottom, it's called the dye-maker's puffball, Pisonia. So it doesn't have the typical bolete growth form, but it is in that order, the boletes, and it's mycorrhizal. And then on the right, there's a brown and a white bolete, the genus Gyroporus. And there's a lot more under oaks. This is the genus Lactarius. So like lactating, a lot of species in this group produce a milk, or latex, if you will, when you cut them. So at the bottom left, it's a little bit hard to see, but I drew an X in the gills, and there's a white exudation that's coming out. So that's kind of the name, the origin of the name. But a lot of the species do not exude latex. So it's not a reliable character necessarily. On the top of the caps, they often have a zonate pattern to them. So kind of a whole bunch of circles on the cap. And you see on the top right, that's a different one in the Everglades I've seen, Yazuensis. It was named for a locality in Louisiana, Yazoo. So it has that zonate form. And then on the bottom right is kind of an atypical Lactarius. It's kind of orangish red, and it doesn't exude latex. It doesn't have much of that zonate color. So it's a very big group, and it's not easy to come up with a character to identify a genus or a family in mushrooms. There's just so much variation. And then back to the genus Russula, we can find them under oaks as well as sea grapes. On the left from the Everglades is a red one. It'll take a lot of work to figure out what that could be. On the middle is a white one. I've seen that one quite a bit. That was in Fuchs hammock. And on the right is sort of a brownish one on the cap, another Russula. So those are Russula, Lactarius, boletes. That's a mycorrhizal group, predominantly mycorrhizal, found with pines or oaks or cockaloba. So now we move on to non-mycorrhizal things. So then that's often what we think about when we think about mushrooms and fungi, or things that degrade wood, dead wood or dead leaves. And those are a few examples there. And that's a big job for fungi. So plants form a complex of molecules called lignocellulose. So it's cellulose and lignin. That's a very tough, complex material that's very difficult to degrade. And fungi are really specialized on degrading that tough tissue of plants. Bacteria do it a little bit, but not really to the degree that fungi do. So that's really one of their big jobs. And this picture from Fuchs hammock kind of illustrates that. So on the forest floor, after nice heavy rain, there's just tons of fungi popping up because these fungi are in the leaves, in the bark, degrading all this lignocellulose. And once the rains come, you actually see the fungus pop up. Often we think of polypores. So that's a group of fungi that you often find on wood, and they're very tough. Right? So they're perennial often, and they might grow for months and months and months. And year round, they might be perennial. And often underneath, at the top left, they have pores, kind of like boletes, but totally different lineage. On the bottom left is Hexagonia hydnoides. That's one of the most common polypores in Florida. Sort of blackish colors, and it's hairy on the top. So if you see that, you should be able to identify that probably pretty easily. On the right is the genus Ganoderma. I often see that on down Bursera, gumbo limbo logs. But polypores can have some beautiful colors. Here's 3 different ones that are orange. On the left is Echinoporia. That's kind of echinate, kind of bristly. And then underneath it has these maze-like fluctuations under its pore surface. In the middle there is something I just found on FIU campus in the mulch. So it's pretty bizarre. It's just on the ground, a polypore just covering this mulch, degrading the mulch, and it's forming pores. And on the right is one just hugging a stick. So it doesn't really form much of a 3-dimensional structure. It's just crustose or resupinate is the term they often use. So it's just hugging the stick, and those are minute pores. It's bright orange. I have no idea what that is. DNA is going to have to help me on that one. And then our typical mushroom shape, they plenty of species that degrade wood or leaves in those. On the left here is Marasmiellus cubensis. I think it's the first report from Florida, but it's turned out to be pretty common. If you go out in a very heavy rain afterwards, after a very heavy rain, you can often see this fungus. And it's only on sticks and logs that are hanging above ground. So it's never on the ground. It's only like slightly elevated off the ground. And they come out after heavy rains, and they're very mushy. So if you pick one or handle them, it just collapses and turns into mush. It doesn't hold its form anymore. On the right is one I found in Key Largo recently, a Vulvariella cubensis. If you look at the bottom of the mushroom, there's a little cup, a grayish brown cup. So that protects it as it's forming, and then once the cap is mature and so forth, it bursts out of that vulva. And then the cap is formed, and then the spores form. And this has a pinkish gills. I was pretty excited to find that one. On the left here is a very bright, nearly fluorescent pink-purple Lepiota. That was just kind of a cool find. I don't really know what more to say about that one, but probably just a grading leaf litter. On the right there is a marasmius. You find these tiny little fungi, so that's maybe an inch high or so, often on leaves. And there's other weird forms. That's part of the peculiarity of fungi. The top left there is Geastrum or earth star. So it's kind of like a puffball again, a different lineage, and it's just usually found in leaf litter and mulch sometimes too. On the bottom left is Penicillium, like penicillin. It's the same genus. And that's just on like dung, maybe rabbit dung or something. So there's fungi specialized on degrading plant material from dung. The center is Cucina tricoloma. So I saw that in Castello hammock. It's kind of a conspicuous fungus. So these very hairy, it's a tough fungus. You can handle it and beat it up. And I've never seen that anywhere else yet in South Florida. So that's very interesting for such a conspicuous fungus. So far, I've only seen it in this one hammock. On the bottom there is something I found at FIU, Trichoglossum. Those are called earth tongues. So it's kind of like a tongue, and it's kind of leathery, just growing in mulch. And it's just a black fungus. It's hard to even know it's a fungus until you kind of work with it and know what to look for as well. On the top right is Pterula, which has this branching structure and grows on degrading wood and leaf. So just some cool things. Stinkhorns are a very odd group. They call these stinkhorns because they often stink. So their spore mass is very sticky, and the mushroom is giving off a stinky odor to attract flies, so that the flies will come to this fungus and pick up the spore mass and spread it around because the spore mass is so sticky. And again, the flies are attracted to it because it stinks. So there's a genus Phallus in the top left, Mutinus bottom left. In the middle there is the genus Clathrus. In the bottom right, there's a coin for scales, a very tiny stinkhorn, genus Laternia, just in mulch. So these typically aren't very specialized in a sense that you can find them in disturbed areas often. So there's some fungi that are parasitic, right? So they're kind of attacking a living organism, such as insects. So these are some cool fungi to find. On the left, at the Deering estate, is Bovaria. So it looks like it's just a fuzzy mold, but that's really what that fungus does. That's its complete look. It's just to make a fuzzy mold on various insects. I think this is a non-native sweet potato bug, I think is what it's called. And it's just attacking those insects. On the right is a even cooler fungus, Ophiocordyceps. So this is a pretty interesting genus because a lot of times this genus, they're specialized on a particular species, often exclusive to a species, like a species of ant or a species of fly here. And they often make that adult insect behave in a strange way where it puts itself out in a very exposed environment where the wind can pick up the spores. So, and this was just my neighbor's yard, but undoubtedly these bottleflies were acting in a way they wouldn't normally act. They were all going to the tips of the tree branches and just hanging out there. And eventually when the fungus kills the insect, it's just kind of stuck and right at the tips of those branches. And it's a great place to disperse spores. So I was kind of excited to find that one. And then there's fungi that are parasitic on plants, even living ones, of course. So the strongback or the strongbark, the genus Borreria, is very common in the hammocks of key largo, or keys. And you can almost identify it by this fungus on the trunk on the right there. So it's kind of a monstrous, weird growth form, kind of a polypore. This genus Philoporia, and it's always on these living trees, and the trees seem like they're doing just fine. So maybe it's commensal in a sense, and not so much parasitic, or maybe there's even a symbiosis that's beneficial to the plant. But it's only on this species, and it's on most of the trees I've seen in the Keys. And it's guessed that it's living off the sap, that these fungi are just living off the sap, as opposed to most fungi degrading wood or leaves, in a sense. So that's exciting. I think that's an undescribed species. Again, a lot of work to describe them. And then I just kind of in line with that is some more fungi that with unknown lifestyles. So fungi that we don't really know what they're doing. So this is a Castillo hammock. This is a sinkhole, right? You're like, what am I looking at here? So there's a fungus on those rocks in the sinkhole. So this is one of my most exciting things for me. I really want to know more about this fungus. So it just peels off of the rock. There is no wood down there that this fungus can be attached to. It's pure rock down there. So something weird is going on. This is the genus Lintnaria, and no one's really figured out what they're doing, because they don't ever seem to be attached to wood, or sometimes they are, but they're usually at the soil level, suggesting there's something going on with plant roots. And I don't even know what it's connected to, because again, it looks like it's just right on the surface of rocks. A very weird thing. So it's bright orange, so I really want to find out what that is. I see a comment here from Lydia. Where is Cucina? Just to kind of backtrack real quick. That's a very Caribbean or neotropical genus. So in Florida, I've only seen it at Castillo hammock. So again, this thing is just found on rocks. I have seen it on above ground rocks, Lintnaria as well, not just sinkhole rocks. So there's something strange going on. Here's the genus Geodyna. Seen this in quite a few hammocks. I thought it was rare at first, but it almost seems common now. And it really had to be very delicate and careful to trace the base of this cup fungus. You see it kind of twists, and it eventually hooks into a horizontal root. So no one had really understood what this fungus is doing, and we still don't completely, but I'm fairly certain it's connected to plant roots. And those plant roots seem to be dead when the fungus is mature, but maybe they're infecting live plant roots, and then they eventually die. I'm not really sure. But this one was from Key Largo, where I really had the time to look at the fungus. Although it was pouring that day, I got absolutely drenched, but that meant also no mosquitoes. But it was pretty dirty that day. The genus Glaziella, it's a pretty weird thing. I've seen a lot of it on FIU campus. It kind of looks just like a wrinkly, sunken orange. And when I first saw it, I had no idea it was a fungus. And I was like, I'm going to pick that up. A lot of times my fungus discoveries are like second guessing myself and like, well, let's check it out. What is that thing? And it is a fungus, just this weird orange wrinkled ball. And it seems like it's just degrading leaf litter or something, but there might be something else going on. It's the DNA work has been done a little bit on it. So we know that it's an isolated lineage, not really related to much else. It's kind of its own thing in the Ascomycetes. And I sent some material to some colleagues, and they're trying to culture it and do a lot more work with it. So we should find out some more about it. And then this is the genus Trechispora. Here it was, we took it back home, and then we modeled it on a sea grape leaf, but it actually just grows in the soil. That's just the best picture we had, just to kind of see that. And up close on the bottom right, so these pinkish bumps. So that's probably where the spores are coming out. So this is the first report for Florida. A lot of the species I've shown are kind of first reports. And I've only seen it once. And it was actually reported for North Carolina about 80 years ago or 90 years ago, and never reported again for the US. And so they do some isotope studies, try to figure out where it's getting its carbon and so forth. And then there seems to be some peculiarities about what it's doing. Like maybe it's getting carbon from plants, live plants, or maybe it's degrading leaf litter as well. So it's kind of a fun thing to try to figure out what's going on. And then here's another one, the genus Phaeoclavulina. I think these are 3 different species in a couple different hammocks. And I only find them in rich hammocks. A lot of these species are true, like this one or Geodyna. Only in really nice, rich, old hammocks do I find these kind of groups. Whereas the Glaziella was just, again, that's just in mulch and things. But a lot of these are just rich hammocks. So are they degrading leaves? Why do they need, these really nice old hammocks? Are they interacting with plants and trees? So that's a big question. Kind of need to figure out. So that's a whole bunch of different fungi I've seen. There's of course a lot more, but I thought those would kind of illustrate what's out there, and why am I studying? And it's just exciting to discover things, of course, for me. But fungi are everywhere. If bacteria can be there, fungi are there. That's pretty much the rule. Fungi are microscopic. They get in, they're on every animal, every plant. And the carbon cycle, thinking about soil carbon, there's more carbon in the soil than there is above ground, right? So those forests, there's actually more carbon below the forest in the soil. And fungi might be forming up to 1/3 of that carbon in the soil through their living and their dead biomass. And biotech, so we can exploit fungi. We've exploited them for medicines. We still use like cyclosporin is very popular fungal-derived medicine to suppress the immune system. That was found just from somebody scooping soil up, basically, and culturing fungi. And we exploit them for many other purposes besides medicines, for other industrial uses. And then I think the more we know about our biodiversity, the more likely we are to care and to conserve it and to find value in it. So we go to Castello Hammock, we can say, hey, we got some rare ferns here. We got some rare animals, some rare snails. Do we have any other rare things? We have rare fungi. We probably have rare fungi there too. So there's more we can say about that hammock and why it's valuable and unique, and why losing it would be losing something that is thousands and millions of years old. These lineages, right? So here's a nice recent study just published earlier this month co-authored by Peter Raven. So he's a very well-known figure in botany in the United States at the Missouri Botanical Garden. And he's just kind of summarizing things we already know from other authors' estimations. But the title is Here Today, Gone Tomorrow. We're losing biodiversity so fast, and fungi are extremely diverse, but we only know about 100,000 or 150,000 or so. We've given names to them, meaning we've studied them in depth. But it's thought that there's at least 2 million species of fungi. So maybe 5% we've kind of studied closely. So there's so much more to find out about fungi. Take the UK, for example, right? They can, at the top there, rare British fungi, right? So they can say something's rare because they've studied their fungi so well. So there's few places in the world that can say something like that. So they report 15,000 species, and it's about, again, the ratio of plants to fungi, 5 to 6 times the number of fungi as plants. And they even red list. They're trying to red list fungi. So here in the title is red list fungi for Great Britain. In the bottom, the genus Cantharellus, species Freesii. And if you go down a few lines, they say extant mature individuals. So they're estimating there's 130 of these chanterelles in the area. Right? So to say how many actually exist, because they've looked for so long that they can say things like this. Or in California, that's one of the best studied US states for fungi. Right? Here's a book on rare fungi. Right? So they looked long enough that they can say whether or not something's rare. So that's kind of fascinating. So hopefully we can get there someday. We can say what's rare in Florida for fungi. So if you want to, document fungi, it's a very serendipitous thing, right? If it rains and it's just the right conditions, you might find a mushroom, right? So those 5 to 10% of fungi that make mushrooms, you might find one. And now that we have cameras everywhere, you can take a photograph. So one thing to realize is if you take a photograph of a mushroom, try to take pictures of every part. So it's frustrating to a mycologist if you take that photo on the bottom left and that's all you give them. You just give them the photo of the top. And that's what's underneath, and that's also important, right? So we have gills underneath, and on the right, I even peeled it apart, and you can reveal this weird staining pattern, this orangish carrot orange staining in the fungus. So those are all helpful characters, and it's good to document these. You can put them on websites, of course, like Mushroom Observer or iNaturalist. Mushroom Observer is devoted to fungi, and iNaturalist is devoted to kind of all organisms. But sometimes you get different feedback and sometimes better feedback on a specialized website like Mushroom Observer. But then I get other users on iNaturalist that help me out and give me feedback. And I should mention also that, so I do collect a lot of these mushrooms, and the idea is that through studies over time that collecting the mushroom, the reproductive part, seems to have little to no influence on the longevity of the species in that area. So it's kind of like taking a reproductive, apple off of a tree. You're taking the reproductive component and the spores, there's millions of spores that have probably already been released. And again, studies so far don't show any negative impact unless you're trampling and degrading the soil. That's when you start to see a loss of mushrooms. And documenting them with collections, like collecting the physical specimen and drying it, that is extremely useful, and that's how we make a lot of progress in understanding fungi. Like you can see the spores on the top right here, that's a picture of spores. Those are important characters, looking at cells, and then the DNA. So the physical specimen can provide DNA, which is very, very useful for studying fungi. It's kind of, they don't make pretty specimens. Like plants, you can press them and flatten them, and they look very nice. Fungi, once they're dried, they lose a lot of their color and shape, but you can still study them quite a bit, and that's important for mycology. And again, reminder, most fungi are microscopic, so we're going to have to dig deep, if you will, and look closely under the microscope and do a lot of environmental sequencing, culture studies, all sorts of things to find out really what's around us, and not just the macroscopic big fungi. And even humans, right? So we could study the mycology of humans. Right? A healthy human has, on the bottom left there, yeasts living on their skin. We all have them. We all have some fungi in our intestines. So just healthy humans. And we're probably all indoors right now. We're breathing in mold spores. It doesn't matter how clean your house is. There's resilient fungi and there's spores floating around. And we could say the same about the tiger beetle. It has fungi on it and inside of it, undoubtedly. So we're conserving the Miami tiger beetle. We're trying to save that, but we're also trying to really save a lot of things that's going on with it. So it's ecological interactions outside of itself, but even inside of itself. There might be endemic species only on that tiger beetle, or there's probably lots of just facultative species that associate with it. But just saving that one species is really saving so much more. And same for plants. You tear open a leaf, there's actually fungi in there under the microscope. In the roots, there's fungi living on them. So there's just so much going on. When we're conserving things, we're protecting more than just what we see. So there's this quote up at the top. My friends made everything in life as not really anything more than fungusy weirdness. So I just thought that was kind of a funny way to say, fungi are everywhere and so forth. That's Aryan Free at top left, graduate student. He's helped me find out a lot about these fungi. He's doing a lot of the microscopy and DNA work. He's at USF. I was there previously, FIU. I'm very thankful for their support. Everglades, Dade County, Dagny Johnson State Park. For their permits, Mushroom Observer, a lot of people on that website have helped me. And the bottom is my family, which I'm very thankful for all of their support. So that's all I got. Let me just look at some of these on the bottom here. I don't know much about fungicide use, to be honest. George's question, but and guided fungi tours that. I mean, I'm just kind of starting to find out what fungi are around us and where they're at. But a lot of these places are very delicate, sensitive places that you can't just get into, just driving up to it at any time. So, I mean, right now I don't know of any fungus tours, but I mean, I'm definitely interested. That's part of why I'm giving this talk, is to have the outreach and to get people excited about it and appreciate and value mycology a lot more. Let's see here. I guess I can stop. No, I can't. Okay, so any other comments or questions?
Hey, Alan, I have one.
Yes.
So so I find it pretty incredible how little attention fungi are given in the broader narrative of conservation, given their integral role in maintaining the health of ecosystems. You can't really imagine a forest functioning as healthily as it can without the fungi that are associated within the ecosystem. So in your opinion, do we need to really start pushing the importance of these overlooked species if we are to have a successful conservation and restoration projects? Or do you think the current route that we're at would, where we kind of continue with conservation and restoration as they're usually done, will still kind of indirectly work on its own?
I'm not sure if it's your what I'm not getting a good signal on, and I didn't hear much of anything you said. And it was cutting in and out.
Oh no,
maybe it's doing the same when
yeah, it's cutting a little bit now with yours too, at least on my end. Can you hear me now?
time for some reason.
Hopefully that works.
Hmm. Yeah.
Can you see it now, Alan?
It's okay.
On the chat.
Yeah, I can see the comments in the StreamYard. It's suddenly it's back. The signal seems back. I don't know. It was really rough there for a little bit. I'm sorry.
Yeah, something happened there towards the end.
So what was your question? You're saying, like, there's a lot of.
It was the one under Dade chapter.
Oh, well, yeah, of course. I'm biased towards fungi. I want to include them in the narrative more. But it's hard because we don't know them that well, so we can't say very well what's rare, what's unique, how important something is. So we have to look a lot closer to say those sort of things. But it is good to realize, even roughly, when we talk about protecting something, that there's an incredible amount of diversity that we're protecting that we have know nothing about. There's lots of protists in these forests. There's lots of fungi. There's lots of bacteria that we know almost nothing about. And there's thousands and thousands and tens of thousands of species in a forest, really. So I mean, I really want to see more work on all these things: microbiology, environmental microbiology, and stuff like that. Because those are questions in my mind I'm just curious about.
Absolutely. Thank you.
Let's see here. do some plant genera have more microscopic fungi than others? So I mean, again, the mycorrhizal groups, they tend to have mushroom-forming mycorrhizae. So like oaks and pines, that you can find a lot of things typically under those, or cockaloba. But there are other associations that might not be mycorrhizal where you find microscopic fungi, but I don't have any good examples off the top of my head, other than, of course, these forests, their leaf litter and all that sort of stuff. Their decaying wood support a lot of particular fungi. So mycorrhizal relationships are pretty well studied in a lot of plants, but again, temperate. So the tropics and neotropics is there's a lot of new information that we're just now peeling away at with mycorrhizal stuff and so on. Let's see here. Somebody mentioned YouTube questions. I don't know if I can see any of those at all or not.
Yeah, if we're seeing them here in the chat box, and then we're getting them, and it seems we're getting the ones from Facebook.
Okay, very good. That's good. A good guide to edible fungi. So that's kind of goes back again to not knowing fungi very well in Florida. So it's hard to say what's edible, what's safe to eat, and what might be safe to eat but might easily be confused with something that's poisonous. So we don't know them that well to make safe safe opinions about those, I would say. The chanterelles, people are eating those, and that seems like nothing that needs to be that's too dangerous. But I don't tell anybody to eat any fungi because it's a tricky game. Even seasoned mycologists have made the mistake, and sometimes that mistake can only be made once. And there's deadly fungi. The genus Amanita, I saw a comment earlier, that has some of the most poisonous fungi in the world, absolutely. But there's also species of Amanita that people eat. So people find and figure out what's tolerable. But some people also tolerate different species than others do. So it's not an easy thing to figure out right now because we don't know them that well. So why do, like, the genus Lactarius, why do they have an exudation? That's a good question. I honestly haven't looked into it that much. And maybe, like the latex and like the Posinaceae, like the milkweeds, right? They can really gum up the mandibles of an insect or trap or prevent them from moving around a lot or eating. So probably the latex has some properties that might help it defend off any things that eat it. That's just a guess. And there are some sometimes we do taste the mushrooms. Back to the edibility thing, a lot of mycologists taste mushrooms and we just spit it out, and it's generally considered a safe thing to do. Again, I don't really advise anybody to do that stuff, but there are some mushrooms that are bitter and some that are even spicy, like a hot pepper, like a jalapeno. So the genus Lactarius, there's a couple species that's kind of a helpful character. You take a nibble and it's like biting into a jalapeno. It's very, very strange. So the Lactarius, in addition to having latex sometimes, maybe like these spicy chemicals also deter maybe some insects or slugs or snails. So I think all the chanterelles are Cantharellus cockalobae because they're always found with sea grape or pigeon plum nearby. So I think that's, in addition to morphology, sometimes morphology is not that helpful for species identification, but the host association in this case, I think, is very exclusive, and it's only the Cantharellus cockalobae. I don't think there's any Cinnabarinus in South Florida. There might be with some oaks, maybe in North Miami or something like that, or maybe in some hammocks. But if you see a chanterelle with cockaloba, I'm fairly certain that's Cantharellus cockalobae. So invasives and exotics for fungi. So that's a hard question to answer, again, because of our little knowledge of fungi locally. So it's almost like we discover something here. We find a mushroom. We have no prior knowledge to know if it was here already or if it's just a new introduction. We can infer that quite a bit because if they're growing in disturbed areas and the rest of the population is all in Australia or something like that, it's probably an introduction. So that has been the case recently. There's this common lawn mushroom that's an orange marasmius. So a lawn, that's of course a fairly recent invention of man in a way, these short grass cut lawns. And it turns out somebody in Australia found these in the wild, in the woods. And Arian started working with them, and the mushrooms turned out to be the same species by DNA and morphology. But here they're in a kind of a disturbed lawn, and then in Australia they're in a woodland. So it's probably they're native there and they're introduced here. But whether any of those are a threat to biodiversity and so forth, that's a very difficult question to answer. There's some orchids that are spreading in the state, like the monk orchid, and orchids depend on fungi. Their seeds are incredibly tiny. They have almost no nutrition, and it's usually a fungus that stimulates their germination. So it could be that some of these invasive orchids are maybe stealing fungi from other native orchids. I mean, this is pure speculation, but there could be things like that going on as well. So I don't know.
Alan, I noticed there was another question by Ariel
Yes.
higher up in the chat.
Yeah.
She asked.
Yes. Photos or specimens? Absolutely. I mean, like the chanterelle, the Cantharellus cocklobe, we pretty much figured out what that is and so forth. We can learn more about its ecology, certainly. So, specimens of that are probably not that important. But again, there are serendipitous things to find a mushroom often. So tons of researchers, benefited from just citizens saying, hey, I found this weird thing, and now that we have iNaturalists and so forth posting on there, definitely, Arian has contacted people and say, hey, you're up in Jacksonville. That's a really weird mushroom. I'd kind of want to know more about that. And he's had people send them specimens, certainly. So it's definitely a useful thing. And posting them on iNaturalist is a great way to kind of start that cooperative arrangement if you find something interesting. Again, natural areas, we got to be concerned about permits and things. Right, so I get permits when I go to these places, but we find cool stuff in our yards as well, and some of us have natural areas in our property even. So those are all considerations. But I do look at iNaturalist quite a bit, so if something's interesting there, there's a good chance I might see it, but I might not catch everything. But anybody is welcome to email me if they see something and they just want my comments or opinion or what it might be or something. But again, it's photography only tells us so much about mushrooms. DNA is telling us so much more than we ever could have imagined. So it's hard to identify mushrooms from photography, especially in an area like South Florida where we don't know them that well. So in the future, we'll know them well enough where a photograph, we can say, okay, I think I know what that is. But there's only a few that we can really confidently identify through photographs. Let's see, I know some animals are similar on different continents. Can we talk about invasives in this case? So that's there are a lot of species that are similar on different continents, but with respect to native fungi, a lot of times we find out that these are different species and they just look the same. So we've been applying a lot of names that were described, like fungi that were described from Europe. It looks the same over here, so we apply that name, and then suddenly we start studying them, the DNA and so forth. We find out that actually that's a totally different species. That we have a native thing, the European thing looks the same, but it's actually different. But I don't know. There are certainly cases of invasives, especially like pine plantations. So they plant pines around the world, pines are very useful for wood pulp and making boards and all sorts of things. So they find a lot of these mushrooms popping up under these pine plantations that, of course, are not native. So that happens a lot at times, carrying the host tree to a new place, and the fungi end up there as well. Introducing fostering fungi growth in our yards. I'm honestly not that experienced with culturing fungi, but people have been successful in many cases just taking the mushroom caps and putting lots of them in a good spot in their yard, and they might come up next year. That's definitely happened. Of course, these fungi, many of them are very particular. So some of them need a specific host tree and need an oak tree. Some of them need a really damp, shaded area, and some of them like mulch. So it's going to be different for every species, but people have succeeded, definitely, of just taking mushrooms and let all the spores fall in an area that they hope the fungus will like. Oyster mushrooms, people culture those a lot, like on newspaper or cardboard and all sorts of things. So there's some mushrooms that are very plastic with their habitat requirements.
I have found too that if you ever come across like wooden logs that someone's tossing, it's good to repurpose, to just leave them in a corner of the yard and just let native spores find their way there. And I've seen all kinds of interesting stuff pop up on them.
Definitely. Okay, I think I got most of the questions.
It seems so. All right, Alan, thank you so much for this presentation. Speaking personally, I know next to nothing about fungi, so this was extraordinarily informative for me, and it seems from a lot of the comments that the people who tuned in feel the same. So thank you so much for being here tonight.
All right, thank you all for attending. I hope it was useful and enjoyable to you. I see some familiar names like Dr. Suzanne Copter. Good to see all you out there. All right,
Alan. Thank you so much, and thanks to everyone for tuning in. Hopefully, you guys enjoyed the new platform via Facebook and YouTube, and we hope to catch you in the next one. Everyone, have a great rest of your night, and take care.
Bye.